In this video, we'll start discussing cell signaling. One property of all cellular life is that it needs to have the ability to interact with its environment and respond to various stimuli. This is true for single-celled organisms, such as this paramecium. Single-celled organisms, even though they only consist of one cell, must respond, first of all, to abiotic factors, such as chemicals in the environment, light, heat, and pressure, as well as respond to interactions with other cells. So you can see here that this paramecium now is interacting with and responding to a cyanobacterium. This looks like oscillatoria that's coming in from the right, and paramecium is going to have to respond to this interaction. This is true even more so for multicellular organisms, such as these beautiful African wild dogs. Multicellular organisms must not only respond to the environment, which you can see them doing here, but they must coordinate the cells within that multicellular organism and mount responses within the organism as well. You can see these wild dogs are radio-collared, so it looks like scientists are studying their movement and behavior. These are one of my very favorite organisms of all time. All right, this brings us to cell signaling, in which cells respond to signals from outside the cell. We can divide signaling into 5 major steps. This is a generic signaling pathway, and we're going to study this as an overview, and then later we can dive into the details. We start with a signaling molecule. We're going to call this signal molecule a ligand. This is a generic word for a molecule that binds another specific molecule, and in biology we most often use the word ligand when we're talking about something that binds to a receptor and initiates a signal. Ligands can be many different types of molecules, including proteins, single amino acids, short chains of amino acids, in other words, peptides, glycoproteins, meaning proteins that have been modified with sugars, lipids such as steroids and others, phospholipids, and even gases such as ethylene and nitric oxide. Some of these molecules will be hydrophilic, which means they're less likely to be able to enter the cell. They may be kept outside in the extracellular space. Others are hydrophobic, such as our lipids, particularly our steroids, meaning they're more likely to pass through the plasma membrane and enter the cytoplasm of the cell. Ligands are produced by what we call the signaling cell. Okay, so we have a signaling molecule that's been produced somewhere, and now it needs to bind to a protein called a receptor in order for the cell to respond to the signal. This diagram shows a receptor that's embedded in the plasma membrane of the cell. We call this a cell surface receptor. Cell surface receptors are transmembrane proteins that bind extracellular ligands. So generally molecules that would have difficulty crossing the plasma membrane, such as a large protein or a charged and therefore hydrophilic ligand. What happens when a ligand binds to a receptor? Recall that when anything binds to a protein, the protein can undergo a change in shape, sometimes called a conformational change, and that can affect its activity and ability to bind other substances. Think of enzymes and the way that they change their shape when a substrate, activator, or inhibitor binds to them. This is similar. Okay, so when our ligand binds to the extracellular domain of a cell surface receptor, this causes a shape or conformational change in the receptor, which is how the signal gets from the outside of the cell to the inside of the cell without the ligand having to actually enter the cell itself. Once the receptor has changed shape, it becomes active, and this initiates a series of events on the inside of the cell. However, some receptors are intracellular and exist in the cytoplasm of the cell. In that case, the ligand would cross the plasma membrane and enter the cell in order to bind to the receptor. That's more likely to happen, of course, when the ligand is a small hydrophobic molecule that's able to diffuse through the membrane into the cytoplasm, such as a steroid. Some intracellular receptors are also transcription factors, and upon binding the ligand, they become activated and can move into the nucleus and bind to DNA, turning transcription of messenger RNA on or turning it off. And receptors, by the way, are specific. They generally bind just a single type of ligand, or perhaps just a few molecules that are similar in shape and chemical properties. Okay, once a ligand binds to a receptor, thereby changing its shape, the message is propagated through a signal transduction pathway. In other words, there are a series of events that take place that eventually result in the cell's response. You can think of signal transduction molecules as passing the message on until it reaches the molecule that needs to receive the signal and can actually do something. There are several ways to accomplish signal transduction. One of the most common is a series of phosphorylation events in which phosphate groups are added to a protein, which in turn phosphorylates another protein, which in turn phosphorylates another protein, and this will continue until the signal reaches the protein that will actually mediate the cell's response. The fourth step in cell signaling is what we just mentioned, the cell responding to the signal. A very common response to the signal, which we've already mentioned, is to regulate transcription. So either turning on the expression of certain genes, such that messenger RNA is made from that gene and ultimately expressed as a protein, or perhaps turning off certain genes such that transcription of those genes stops. That's a pretty common response, but there are others. Responses can also include increasing metabolism, cell growth, apoptosis, and there are others as well. Finally, the last step is termination of the signal, and this is important. The cell doesn't necessarily want to keep the response going indefinitely. That would probably cause some sort of chaos in the cell. The response needs to be proportional to the signal. We don't want to just, for example, turn on a certain gene and have it active forever and ever. So the cell may degrade the ligand and or degrade or inactivate components of the signal transduction pathway such that the response to the signal ends. And that is our overview of cell signaling. Cell signaling can get pretty complex. There's a bunch of different pathways, different ways that cells can receive and respond to signals. But this is the take-home message. So keep this in mind when we're talking about any type of cell signaling.